Good morning, everyone, and it is my pleasure to present our recent work on the potential interaction between calcite dissolving bacteria and the soil calcium cycle. And as most of you know that calcium is essential nutrition for pod development for peanut. And when there's not sufficient calcium, the peanut can develop a lot of defects, including embryo abortion, forming a structure like a pod without embryo called pops. And there's some recent studies showing that a low calcium level in soil is associated with high incidence of soil fungal pathogen. For example, the Aspergillus flavus, the causing agent of aflatoxin contamination. The importance of calcium for peanut lays in the concentration of calcium in the packing zone. Some very nice work done in the 1940s showed that developing pods directly absorb calcium from its packing zone soil. The calcium in developing pods are not transported from the root system. In this figure on the lower right side showing that these two plants are growing in full nutrition. The roots of these two plants are growing in full nutrition, but the packing zone for the plants on the left lacks calcium. As you can see that although these two plants have very nice vegetative growth, the plants on the left without calcium did not develop enough pods. So demonstrating the importance of calcium in the packing zone. And in Georgia, for example, if the concentration of calcium in packing zone falls below 500 pounds per acre, then adding gypsum or limestone calcium carbonate is recommended. And for peanuts that are saved for seeds, actually gypsum is recommended for all these plants. In the recent years, actually in the last 2 decades, people start to realize how important the microbial community in soil are for plant growth. And this microbial community known as microbiota or microbiome plays critical roles for plant physiology and development and resistant biotic or abiotic stresses. And one aspect of the importance is that these microbes can help plants to absorb various nutrients. Interestingly, one group of bacteria, actually they're present in a diversified taxonomy. This group of bacteria can dissolve insoluble limestone, for example, the calcium carbonate, into soluble calcium and eventually increase bioavailability of calcium in soil. And here I'm showing you one example of a calcite dissolving bacteria, also known, we call it CDB, on a plate. In this plate, this calcite dissolving bacteria creates a halo because it dissolves insoluble calcite added into this plate. And this group of bacteria have been found in calcite-rich environment, for example, the coral reef, limestone mine, and shells. For example, these are oyster shells. People believe that these bacteria have a function to dissolve this calcite on these shells. And for our research, we are interested in these questions. For example, the first question is, are there calcite dissolving bacteria in peanut field? And what are they? And the second question is, can these calcite dissolving bacteria in field actually alter calcium level? And the third question is, what is the dynamics of CDB population in the field, especially in between the CDB population and the calcium level in soil? To answer the first question, Alan Pepper, the graduate student working on this project, he gave a presentation 2 days ago, working on the packing zone soil from the experimental field called Black Shank Farm in Tifton, Georgia. And he used this growth medium supplemented with 1% of calcite and successfully isolated 65 strains of calcite dissolving bacteria. And here it shows you an example of one calcite dissolving bacteria isolated from the plate. And we assign a solubilization index for each CDB. And a higher solubilization index on the plate means this bacterial strain has a higher ability to dissolve calcite. We further sequenced the 16S rDNA of all the 64 strains and identified 17 unique 16S rDNA sequence and grouped these CDB into 10 genera. And as you can see from the list that this is a very high diversity of bacteria from different genera. And this is a very interesting list for us because when we compared the calcite dissolving bacteria reported from two previous groups, including the Rana et al. 2015 and Tamir Silva et al. At 2018. In these two groups, they've isolated CDB from cow waste and a limestone mine, and they found that predominantly a Pseudomonas and Bacillus in their environment. And this is very different from what we found in the field with peanut growing history. And this raised the importance on the value to isolate local and native calcite dissolving bacteria for potential application because calcite dissolving bacteria from other locations may not survive in this environment. Next, we ask an important question, whether changing the level of CDB can alter calcium level in soil. So Alan did two mix. One is we called all CDB mix. This is a mixture of 17 unique CDB strains. And a second mix is we call it top index. This is a mix of 7 CDB with highest solubilization index. Compared to the control condition, which has calcium level around 170 ppm, adding all CDB or top mix at a low concentration at OD 0.01 did not change calcium level. But if we increase the concentration to 0.1, both of the mix showing an increase of calcium level in soil. Then we move to individual strains. Of all the 14 strains that we have tested, 6 individual strains, once they're mixed in soil at a certain concentration, 6 of them increase calcium level larger than 10%, and 6 strains did not cause any change. The range is in between 10% to minus 10%, so we assign them no change, and 1 strain actually reduced calcium level. And I should mention that, as Alan mentioned in his presentation, actually none of these strains cause negative impact on seed germination or early seedling growth. And we also find that although we assign solubilization index to this individual strain for their ability to dissolving calcite on plate, the solubilization index actually is not a good indicator of their ability in soil. In other words, the higher solubilization or high increase of calcium in soil does not associate with a high solubilization index in plate. The next question we ask is, what's the dynamics of CDB population in soil, especially in correlation with calcium level? And this comes to start with experiment Alan did by comparing the calcium level and the population of CDB in different type of soil with different crop history. And he found an interesting negative correlation that fields with high level of calcium tend to have low level of CDB population. And because these are field soil with different plant history, and calcium, it's an interesting correlation, but maybe there are other factors. So Alan further looked into this interesting observation by artificially adding gypsum into one type of soil sample. And this creates a gradient of calcium from about 200 ppm to 3000 ppm. And there's still a negative correlation between the CDB population and the calcium level. So we think that there may be a natural response in terms of bacterial population to calcium level. At a low calcium concentration, it's either created a environment that favors growth of calcite dissolving bacteria, or the increase of calcite dissolving bacteria tend to balance the calcium level in soil. With this, we are working on some future plans that will further our understanding of the role of CDB in soil. We're determining the impact of CDB on pod growth in low calcium soils. And we're also investigating the biodistribution of CDB. We want to know whether these CDB are associated with pod development or root development, or just in bulk soil. And Alan is also tracing the persistence of CDB in soil. So Alan created some bacterial strain that carries about resistance to antibiotics, and this enables us to trace the amount of CDB that we added into soil. With this, I would like to thank people who contributed to this project. Alan Pepper, who is a very talented and hardworking graduate student. All these data are from Alan. And I would appreciate collaboration from Dr. Tim Bradman, and he helped us with all the field work and give us a huge amount of information on peanut physiology. And we also like to thank undergraduate students, Nicole, Annie, and Aisha, who worked on this project. And the funding source of this project are from Georgia Peanut Commission and the startup funds from University of Georgia. Thank you very much. I'm ready to take any questions.
Thank you, Dr. Yang. We've got time for several questions. If there are any, I'll take a look in the Q&A part of the toolbar and see, or the chat. I don't see any questions. Okay. All right. Thanks a lot for your presentation, and all the hard work, and we enjoyed seeing that. Thanks a lot. Mm-hmm. Okay, actually, Lee, we do have one question, and we got a minute or so we can address that.
I'm still here.
Okay, all right. Did you look for CDBs in the root zone? And that's for Mark Hopkins.
Oh, we haven't looked for CDBs in the root zone. One of our plan is to systematically look for CDB population in the root zone, and actually soil associated with root, and maybe some of the CDBs are endophytic. So that's kind of the next stage of the plan.
Okay.
Thank you for the question.